皆さんこんにちは。UTEC がお送りするポッドキャスト番組テックスタートアップの舞台裏、パーソナリティの UTEC Iinuma です。この番組ではテクノロジーやサイエンスに強いシードアーリーフェーズのスタートアップを中心に投資活動を行う UTEC が毎回起業家をゲストに迎え、ベンチャーキャピタリストとともに事業の裏側を語っていきます。今回は前回に引き続きゲストにEditForceの小野社長をお迎えし、 UTEC のプリヤンカとともにトークしていきたいと思います。さて、EditForceはですね、より海外のオーディエンスにもたくさん知っていただきたいということで、後半は英語でお届けしたいと思います。Hello and welcome back to UTEC's podcast. We are thrilled to have Ono-san, CEO of EditForce, with us again today, alongside with Priyanka, our senior associate capitalist. In the first half, which we recorded in Japanese, we discussed the biotech industry as a whole and Ono-san's career journey.Now, for our international listeners, we will be switching to English to dive into EditForce business, what they're trying to tackle, and why it matters. So, Ono-san, thank you very much for joining us again. To get started, could you please reintroduce yourself and give us a brief overview of EditForce?
Sure. Hi, everyone. Thank you for watching our podcast. I'm Takashi Ono, the CEO of the EditForce. I joined the EditForce in 2018, and then I'm leading now the company from 2019. My background is molecular biology, and then, I'm now very, enjoying the journey with the EditForce. EditForce itself is founded in 2015 in Fukuoka Prefecture. And we have now around 20 employees and focusing on RNA targeting therapy. Thank you.
Priyanka, can you give us an introduction?
Yeah. Hi, everyone. I'm Priyanka. At UTEC, I mainly do bio healthcare investments. I am a molecular biologist by training. I previously worked in the cell and gene therapy industry, and I'm really excited about what EditForce is doing here. Yeah, so EditForce is a gene therapy company, and but what they're doing is really interesting. UTEC invested in EditForce close to 10 years ago, before RNA editing was trendy. EditForce was doing RNA editing when this was just coming out. Everybody was talking about it, but no one was doing this. So EditForce has a very unique approach to RNA editing that Ono-san, you can explain later. Maybe can you talk us through the PPR platform that is the backbone of EditForce?
Sure. So let's show our first slide. After this is the PPR, our very unique base technology. The PPR itself can recognize and bind to the RNA. This is quite unique, the technology. Nobody can do the directly targeting RNA at this moment. So we focus on this molecule, develop it, and then get a very nice pharmaceutical product in near future. The PPR consists of 35 amino acids. That's one unit. And this unit in this slide is called PPR protein. The left side, yellow one, is one motif. And right side, pink one, another motif. One motif can target one amino acid.
So EditForce has a PPR technology as its platform. And this is very unique because first, it comes from plants. And what this technology is that one PPR protein, which is made up of 35 amino acids, one of this motif recognizes a single RNA base. And you can line up these multiple PPR proteins to make a single polypeptide. Also, you can attach effector domains at the end of it to do other functions that I'm sure Ono-san can talk about it later. But that way, we can customize the RNA sequence that this polypeptide recognizes, plus with the effector domain, add functions to it, such as RNA editing. And this technology can be used to address multiple genetic disorders that arise from RNA repeats or other RNA-based genetic disorders that are difficult to treat with other modalities. And this technology, PPR technology, is very unique. It comes from Kyushu University. Maybe Ono-san, can you walk us through how this technology was discovered and how EditForce was founded?
Well, I'm the Professor Nakamura in Kyushu University, and our CTO, Yagi, found this molecule from the plant, as Priyanka-san said. And then they tried to, find the mechanism of recognition of RNA by this molecule. And then they got the IP with Kyushu University. At the time, and our founder, one of the founder, Kisco, is looking for the good technology of RNA targeting technology. So they found it in the University and they decided to start EditForce. That's our history.
Thank you.
I'm sorry, it's a very basic question, but what kind of like disease you're targeting for this by this technology?
At this moment, we have one lead product targeting for the DM1. So maybe next slide show you some information. Oh, this one. Okay, so DM1 is a myotonic dystrophy type 1, very rare diseases, and then we don't have any good therapy at this moment. So high unmet needs there, and also this is an orphan, diseases. We found our one molecule, PPR, can target this DM1 diseases. So we started the development in 2018, I think. And then now, under the preclinical studies, and we are getting IND very soon.
But why is it was it difficult for others to treat this disease in the past?
As Ono-san was mentioning, so there is a gene that we have called DMPK. And in the patients that have this disease, there's multiple CUG repeats, sometimes 50 times or more than 1,000 times more CUG in this gene. And what this causes is that RNA splicing errors happen, and that leads to muscle not being able to be relaxed. So that's why you see myotonia, and the patient, you see cardiac issues. There's neurological developmental issues, and this although it's a rare disease, it affects thousands, tens of thousands of patients globally. So although it's rare, it affects a significant portion of the population. However, DMPK as a gene, that is something that we need in wild type. What we can do with EditForce's technology is that because it targets mRNA and not the actual DNA, we can have the PPR protein bind to the multiple CUG repeats and release this MBNL factor so that mRNA splicing errors are reduced. That's the mechanism behind EditForce's lead program, EF210.
Just for additional explanation for Japanese listeners, this disease is in Japanese called kin-kyochokse dystrophy, right? Isn't it? Yeah.
So I think that this mechanism action is very unique, and then this is a quite an important point for us to distinguish, our technology from the others. So others, cannot do like ours. So our other technology may be, using siRNA or something antibody to, compete the DMPK or et cetera, et cetera. But as Priyanka-san said, it's quite, risky. In terms of, the safety. So we believe we can have quite a nice positioning among others.
Like regarding the business, what is the current situation and how has been proceeding so far?
As I mentioned earlier, we are doing still the preclinical studies, but it's final stage, I think I can say. So we already had a good discussion with PMDA, Japanese authority, and also FDA, American US authorities. And we can have now the clear vision to go to clinical trial. So we are preparing IND in next year. So we are, finalizing all preclinical study at this moment. Also, we are doing manufacturing, the process as well. So, very now the encouraging, very important, time at this moment.
The feedback from the FDA was very positive. The clinical plan, the non-clinical data, there was very little negative things that they had to say. There were just minor housekeeping items that the team has to take care of before they can submit their IND. But it was very encouraging to see the positive feedback from the FDA. And the PMDA is also very supportive in the feedback that we've received.
So I was going to ask that I think the UTEC has our first investment from UTEC was back in 2015, maybe. So
yeah.
Also, do you have any like episode that when we
we have a lot. I should say, I can say, every time UTEC, of course, as investors as well, but especially UTEC support us. they gave us the advice, not only plan, but also advice or, view of the, direction to us and guide us. So we are very, happy to be with UTEC and to, develop the product as well, company.
And from UTEC's perspective, I mean, we saw the potential of RNA editing very early. We saw the unique aspect this technology has. The team is very strong. And we can go into a little bit later, but EditForce's technology, the platform, can do gene editing, RNA editing that is not possible with other technologies. So that's quite unique.
Priyanka, you joined the UTEC last year, right? And
yeah,
and then how did you get involved in with the EditForce right now?
Yeah, I've been involved with EditForce for about a year now, and I was very charmed by Ono-san's optimistic attitude and the dedication that the scientific team has to develop this technology. And I think that the PPR technology fills a need that is not being met currently, even with the current medicines that are in development. So we believe that the EF210 can fill that crucial gap.
And Ono-san, what do you think is the VC like UTEC can add value to drug discovery startup? What do you think about that?
Well, big for us, of course. And of course, the science part, we can discuss, the part, and then, also the strategy, how to, contact with the pharmaceutical company in overseas, etc., etc. Moreover, it's important, I have the good, advisor beside me. So not only science, but also, business side or sometimes, personal thing as well. It's easy for me to contact with UTEC and then get their advice. This is quite important for the company and also for me as well.
Any comments, Priyanka?
Yeah, thank you, Ono-san, for saying that. UTEC tries to add value wherever we can, and there's many aspects that a young startup needs help, and that can mean that bringing in scientific advisors that can provide an outside perspective, BD experts, introducing potential partners or other VCs. But I think it's often just having somebody to listen to, right? Being a sounding board. So we're always here for, so I always pick up Ono-san's phone call. You can call me anytime. So,
of course, sometimes, very headache, some hard word from the UTEC or VC, but that's that, the VC is VC and then the company is company. So, we both of us, going the very bright future. So, discussion is very important.
Thank you. One of the other things that I wanted to ask to Ono-san today is that I heard that you challenged to, you tried to transform the organization after you became a CEO, I think. And then I wanted to ask that what was what challenges do you face in like shifting the organizational culture? And from I heard it's from originally a research-oriented company, but now it's shifted into more like industry-focused company. So what were the key turning points and what was the process?
Well, yeah, the EditForce was a very academic company when I joined. So, my mission is changing to the business-driven company.
Right.
So, as you said, yeah, there are so many, hurdles to change the company, especially the mindset of the, employees, because they love the science, but just love science. They don't, they didn't think about business at all. So I should tell them, of course, science is very important, it's base, but we are the company. And then, the investor invested us to have some, good, result. So, I told employees every day, we should do this way, this way, this way. Of course, again, the science is very important, but the end, we need to think about the business. So it took very long time, maybe 1 or 2 years. I hired some new person from pharmaceutical company or biotech industries. and then, changing some company, the rule, etc., etc. Still, some, part of my company is academic driven, but that's also important. So I leave it, but difficulty is balance. Business is sometimes very important, but still science driven needed. So still I'm struggling sometimes, how to balance, both business and science.
Thank you. Yeah, I think the organization is something like can be like evolved and changed like all the time. So I think like every organization has like every all those kind of challenges all the time. So yeah, and then I totally agree that the balance is more important thing.
My luckiness is a previous CEO of the company, Professor Nakamura. He has some business sense, even he's a professor. So he understands some of the business side thing, and then agree with me to go there. So I have some company difficulty there. Professor and the business side, cannot match. But for our case, that part is not difficult. So that's lucky.
I see. Maybe that's why the EditForce organization has like a foundation for the growth. Thank you. So maybe we want to next we want to talk about like the future or the future plan about of EditForce. So is there any like a business plan going ahead for EditForce, Ono-san?
Yeah, of course, R&D is a very big impact on our company. So we are doing the best effort for EF210 development, but also we have another technology, not only the EF210, of course. So we are developing the new technology as well. So maybe previous slide, Priyanka, what do you think? We need to consider base editing thing or?
Yeah, maybe so here you can explain. So EditForce has a second program, which is an RNA editing program, and this is based on the core modular aspect of the PPR technology, right? So we can change the effector domain, and the effector domain can be switched to an RNA editing enzyme. And this is also a unique discovery made by EditForce. This editing enzyme can do not only C to U edits, but U to C edits. And how that happens. So a lot of RNA editors that are currently on the market or are widely researched, they're ADARs, and they do A to I, or adenosine to inosine, which is read as guanosine or G. And you can do this with an antisense oligonucleotide, or you can do the same type of edits using guide RNA and a CRISPR-Cas system. However, with EditForce's technology, it's a single polypeptide with the effector domain. And the effector domain, it has an RNA selecting the PPR sequence as well as the editor. And this domain can make C to U edits and U to C edits. And there are several indications that the company is exploring right now in applying this unique RNA editing ability.
So for this technology, we had many contacts from the company who are handling the editing A to I or A to G. So they are very interested in the our unique technology C to U, U to C. So we believe we have some good deal with them with this technology, and then get some good future.
We're almost out of time. Is there any additional like topic or to cover or additional comments from you two?
So at this moment, we are looking for some the partnership about the EF210 because we are still small company. So we believe, if we can get a good partner who has the experience in the clinical trial, I think that's a faster way to get the pharmaceutical product. So again, we are looking for the good partner. Also, as I said, this unique PPR editing, RNA editing, we would like to also have a partner as well.
Thank you, Ono-san. Priyanka, do you have any comments?
Yeah, I think the potential of this platform is wide. One advantage is that this is plant-based, and EditForce's, there's internal data that confirms that PPR has limited immunogenicity, right? And this is really important when right now gene therapy has a big safety concern. I think some of that concern is overblown, to be honest, but that still needs to be addressed. And this plant-based approach helps addresses a portion of that. Also, it's truly a platform. EditForce has a high throughput sequence generating and validation method so that the PPR proteins, the polypeptide, can be designed to recognize any sequence that we want and quickly verify the binding affinity. So that way we can select the polypeptides that have the highest affinity. Plus, by switching back and forth effectors, there's a lot of diseases that could be targeted. So I really hope that the company can go out and build partnerships to expand what the platform can actually do.
Thank you very much. Well, thank you very much, Ono-san, for joining us today. That's it for today for today's episode with Ono-san of EditForce and Priyanka from UTEC. And I hope you enjoyed your conversation and gained insights into EditForce's mission in bioindustry. Well, thank you so much for listening and stay tuned and we'll see you in the next time.